So good evening everyone. So I'll be discussing regarding MET protein overexpression in non-small cell lung cancer. So coming to the MET alteration in non-small cell, so basically it is divided in 4 groups: MET exon 14 skipping mutation, MET amplification, MET protein overexpression, and MET fusions. So what are the clinical significance of each of these? So MET exon 14 skipping can be seen upfront in treatment naive patients as well as a resistant pathway after EGFR and ALK-TK exposure in respective patients. And we have whatever the available TKI like capmatinib and tepotinib, these are most effective in this subgroup of the patients. Then next is the MET amplification. Again, MET amplification is commonly seen as a resistant pathway after exposure to EGFR and ALK-TK. Rarely 1 to 5% of the cases of MET alteration present as upfront MET amplification. Again, these subgroups are generally considered little resistant to the available MET TKI like capmatinib and tepotinib, though they can be effective, but the magnitude of benefit is significantly lower as compared to the exon 14 skipping mutation. The next is the MET overexpression. These are generally seen upfront and also can be seen as a resistant pathway after exposure to different TKIs. Overall, the MET overexpression till few years back there was no any effective therapy, and these are just considered as a poor prognostic marker. Last are the MET fusions, which are very rare. 0.2 0.3% cases can be seen in non-small cell lung cancer, but these are TKI sensitive group. If these are positive, definitely they can go ahead with the starting TKI as upfront therapy. So today's we will be discussing regarding the MET overexpression population in non-squamous EGFR wild type non-small cell lung cancer patients. So these are results from LUMINOSITY in Asian subgroup of the patients. So basically, Telisotuzumab is antibody drug conjugate where it has a payload in form of MMAE, which is a monomethyl auristatin E, which is a microtubule inhibitor cytotoxic agent. So approximately 25% of the patients which are non-squamous EGFR wild type NSCLC express this MET protein by ISC. And the significance is that these are considered as a poor prognostic group, lead to deterioration of the overall survival in globally, including the Asian race. In the LUMINOSITY trial, which is a phase 2 trial, 2-stage, in the stage 1 they identified the optimal subgroup for C protein overexpressing non-small cell population to treat with Telisotuzumab. In the stage 2, which is expansion group, they studied the efficacy and safety population. So overall, this is the study design, where you can see that they included patients who are more than 18 years, who had advanced metastatic NSCLC, and they had C MET protein overexpression. This C MET protein overexpression was defined as a high or intermediate. So any NSCLC patients who are having more than 25% expression of MET protein by ISC and having a 3+ staining will be considered as a MET overexpression. And out of those who are having a more than 50% of ISC staining will be considered as a high, and between 25 to 49% will be considered as a intermediate expressor. And these patients included those who had received at least 2 lines of or less than 2 lines of therapies. So in the stage 1, they included different subgroup: non-squamous EGFR wild type, non-squamous EGFR mutated, and squamous. So in this, they found non-squamous EGFR mutated and squamous was not eligible for further testing. Only in the phase 2, they continued with the non-squamous EGFR wild type. The primary endpoint of this study was overall response rate, and the secondary disease control rate, duration of response, PFS, and OS. So this is the demographic of overall population, where almost 70% were male population, around 30% were Asian population, and in 20% population had upfront brain mets. MET overexpression in high and intermediate were approximately 50%, and almost treatment details, you can see that almost 97.5% of population received platinum-based therapy, and around 80% population has received immunotherapy drug. So coming to the first overall response rate, so overall there was around 28.6% patient has shown response in this population, which is around third line or second line, And between these two, if divide, CMET high expressor has shown 34.6% response, and intermediate group has shown 22.9% response. And these responses were durable in the range of 8 months, and at more than 6 months, those who had response had a durable response in the range of 56%. Again, those who had high expression had a higher rate of durable response, 63%. And coming to the side effects, so common side effects were peripheral edema, sensory neuropathy, fatigue, and in some cases ILD. So coming to the out of these the Asian subgroups, so out of these 150 patients, 160 patients, there was 57 patients who were Asians, and out of these only efficacy was available in 48 patients. Again, in these 48 patients, around 15% cases had brain mets, and treatment details, again you can see that almost majority of the patient has received platinum based or immune check inhibitor therapy. So this is the patient disposition. Almost treatment median duration of treatment was 4.7 months. Those patients who discontinued drug, almost 50% patient had progression, plus 2 patients has clinical progression, and 3 patients had withdrawn the consent. And among the patients who discontinued study, almost half of the population was died. So this is again response rate in Asian population almost similar to the global population with 35%, and those who are having higher expression had 46.2% response rate. This is the PFS, so almost it was in third line, around 5.5 months was the PFS, and the overall survival was in the range of 17 to 18 months. And those who had a higher expression had a longer overall survival with 25.4%. Again, the toxicity was similar across the globe in Asian population, with the most common side effects were peripheral neuropathy, pneumonitis, and fatigue. So conclusion, so treatment with Telisotuzumab has shown similar outcome in Asian subgroup, and the side effect profile was manageable and similar to global population. And for this, further phase 3 study is going on, where they compare Telisotuzumab with the docetaxel. Thank you.